Good afternoon, class. So today's online lecture is going to deal with Chapter 15, Special Senses. So the special senses are smell, taste, sight, hearing, and equilibrium. And we're going to talk about those in this lecture. So we're going to start off with the eye and vision. So 70% of all sensory receptors are in the eyes. Each eye has over 1 million nerve fibers. So in protection for the eye, you have the bony orbit that the eye is enclosed in. You also have a cushion of fat that surrounds most of the eye. Accessory structures of the eye include the eyelids and eyelashes, conjunctiva, so that is the mucous membrane that covers the front of the eye. Essentially, when someone gets pink eye, it's the conjunctiva that's infected with pink eye. So that's what that mucous membrane that covers the eye, that's what's infected. The lacrimal apparatus, so that's going to be responsible for your tear formation and then the drainage of the tears. And we'll talk about that more in the next slide. And then the extrinsic eye muscles, 6 eye muscles, which we'll talk about also. All right, so this slide demonstrates, if you follow the arrows, from tear formation from the lacrimal gland. All right, then you have the excretory ducts of the lacrimal gland, so the tears are going to leave the ducts, all right, flow over the eyes, and then they're going to drain. First, they're going to go through the lacrimal canaliculus, all right, these small little ducts right here, and then the lacrimal sac. It's going to go down that, and then you end up in the nasolacrimal duct. Okay, so that is pretty much the flow of your tears. All right, so function of the lacrimal apparatus: protects, moistens, and lubricates the eye. All right, so remember, anything in red, always pay special attention to that. So empties into the nasal cavity. So your tears contain dilute salt solution, mucus, antibodies, and lysozyme. All right, lysozyme is an enzyme that destroys bacteria. So there are 6 extrinsic eye muscles that attach to the outer surface of the eye, and essentially um, use the name to help you remember what the actions of the eye muscles are. So if you remember, rectus means straight. Okay, so pretty much when you see um, whatever's before the word rectus, it's telling you the motion, it's action essentially. All right, because rectus attached straight to the eye muscle, so lateral rectus, it's going to move the eye laterally. Okay, medial rectus moves the eye medially. Superior rectus elevates the eye. Okay, and turns it medially. Now, and then the inferior rectus depresses the eye and turns it medially. Now, with the oblique muscles, if you remember, oblique muscles, they attach at an angle. Okay, so they're not attaching straight; they attach at an angle. So their name actually is going to be opposite of their action. So inferior oblique is going to elevate the eye and turns it laterally, and then superior oblique is going to depress the eye, turns it laterally. And you're going to see in the picture how they attach at an angle, and then you can see how why the inferior oblique elevates and the superior oblique depresses. When you look at how they're attached, it makes sense. And then the nerves that control the actions of the muscles. So lateral rectus controlled by cranial nerve 6, the abducens nerve, and then um, superior oblique is controlled by the trochlear, cranial nerve 4, and then the rest of the muscles are controlled by cranial nerve 3, oculomotor nerve, cranial nerve. So if you just remember, um, cranial nerves 6 does the lateral rectus, and then superior oblique is cranial nerve 4. Then the rest of them are cranial nerve 3. So if you remember those two, then then the rest of them are cranial nerve 3. All right, so again, this is just a diagram showing you the muscles attachment. So you see again inferior rectus, you see the muscle and where it attaches. Um, Lateral rectus, you see where it attaches. Okay, and then you see how the inferior oblique, you see this how it attaches, this angular attachment. Okay, and that's why when this muscle contracts, it's going to move the eye up. Okay, and then you see the superior oblique. Um, you see how it goes from superior oblique runs here and then goes through the superior oblique tendon and then attaches right there at that angle. Okay, and so when that muscle contracts, it's going to depress the eye, and then you see the superior rectus muscle, and that's just attached straight. All right, that's going to elevate the eye. But as far as the oblique muscles, if you look at their attachment and then you imagine how what they're going to do when they contract, you see why the inferior oblique elevates, and you see why the superior oblique depresses. Okay. All right, so layers forming the wall of the eye ball. So you have the fibrous layer, which is the outside layer, the vascular layer, which is the middle layer, and then the sensory layer, which is the um, inner layer. All right, so the sclera is the white of the eye. All right, so the white connective tissue layer. So you see the sclera anteriorly as the white of the eye. The cornea is the transparent central anterior portion, allows the light to pass through. It repairs itself easily. The only human tissue that can be transplanted without fear of rejection is the cornea. So that's a interesting side note. The choroid is a blood-rich um, layer in the posterior of the eye, so the pigments prevent light from scattering. And then you have a modified anteriorly into two structures: the ciliary body, smooth muscle attached to the lens, and the iris, which regulates the amount of light entering the eye, and the iris is what gives the eye its color. Okay, and the pupil is just the round opening in the iris. So this slide allows you to see the structures in the diagram. So the ciliary body right here, you see attached to the lens, cornea, transparent, um, front part, anterior part of the eye, the iris right here, that gives the eye its color, the pupil, just the opening in the iris, the sclera, eye, when you go look at the front, it's the white of the eye, sclera, choroid, you see in the posterior part of the eye, it's the nutrient rich layer um, whose pigment prevents the light from scattering. Then you have the retina, okay, which is going to be in the inner um, part of the, so it's going to be an inner eye. It's going to be where the um, your rods and cones are going to be located. Then you have your optic nerve, and essentially your optic disc is going to be your blind spot. Okay, that's essentially it's going to be right in front of where the optic nerve is going to be exiting. Okay, so that's essentially why it's your blind spot. There aren't any receptors in that area, so the optic disc is the blind spot because it doesn't have any receptors. All right, so the retina contains two layers. So it has what's called photoreceptors. All right, so we have rods and cones, and the way you kind of remember is that I think of cones, I think of C for color. Okay, so cones allow for detail, color vision. Rods are perceptions in the gray tones, but your rods are going to be what you need for night vision. This is what you're going to use for night vision. In the figure, you can see where the optic nerve is entering the eye. You can see the optic disc, and then you can see the pigmented layer of the retina. So that's going to be attached to underneath, going to attach to the choroid, and so the pigmented layer of the retina is going to provide the nutrients to the retina. And then you have the neural layer of the retina where you have your photoreceptors, going to be your rods and cones are going to be there. Your bipolar cells and ganglion cells are in the neural layer of the retina. So here's a another figure of the retina. So you see right here the pigmented layer, and then you see right here a rod, which is a photoreceptor, and then you see a cone photoreceptor, and then you see the bipolar cells, and then you see the ganglion cells. So you can see from this figure how light is going to enter this way. So light is going to have to pass through the ganglion cell layer and then the bipolar cell layer to reach and excite the rods and cones. Okay, so then after they excite, after the rods and cones get excited, it, they're going to undergo changes in their membrane potential that influence the bipolar cells that they are next to. And these in turn, the bipolar cells are going to in turn stimulate the ganglion cells. Okay, and then the ganglion cells have axons that lead the retina and tight bundle fibers known as the optic optic nerve. Okay, so the ganglion are going to essentially have axons that are going to form the optic nerve. Okay, and then that goes to the brain. All right, so the lens is a biconvex crystal-like structure. It's held in place by a suspensory ligament attached to the ciliary body. All right, so you see the ciliary body right here, and then you see the suspensory ligaments attached to the lens. Okay, so you see that right there, and you see the lens. All right, so you see how those attachments take place. And remember, always pay special attention to what's in red. All right, so um, talk about some of the pathology as far as of the eye. So cataracts result when the lens become hard and opaque with age. So vision becomes hazy and distorted. It eventually can cause blindness in the affected eye. But you can get cataract surgery, so cataracts can be removed. Risk factors include diabetes, frequent exposure to intense sunlight, and heavy smoking. All right, so here's an image of cataract. You see how if you have something like that, you won't be able to see. So you have two chamber segments of the eye. You have an interior segment, aqueous, all right, and which is interior to the lens and contains the aqueous humor. And then you have a posterior segment, vitreous segment. So posterior to the lens contains the vitreous humor. So so you see right here in red, the anterior segment has the aqueous humor in it. So the lens is in the middle. So you see anterior to the lens, you have the anterior segment, and it has aqueous humor in it. And then you see posterior to the lens, you have your vitreous segment, which is going to have vitreous humor in the posterior segment. Okay. So the aqueous humor is a watery fluid found between the lens and the cornea. So it's similar to blood plasma, helps maintain intraocular pressure, and it provides nutrients for the lens and cornea. The vitreous humor is a gel-like substance posterior to the lens, so it prevents the eye from collapsing and it helps maintain intraocular pressure. So, um, pathway of light through the eye. So light must be focused to a point on the retina for optimal vision. So the eye is set for distant vision over 20 feet away. Accommodation is when the lens changes shape to focus on closer objects less than 20 feet away. So you see the light coming in and you see how it gets focused at one point on the retina. All right. And you can also see how the lens is accommodating depending on, you see light from a distance source, you see the shape of it. And but then you see how when you get light from a near source, see how the lens is accommodating so that you still get that focal point on the retina. So again, you see that light from the near source, you see the lens is accommodating, so you still get that focal point. Okay. All right, so eye reflexes. So internal muscles are controlled by the autonomic nervous system. Bright light causes pupils to constrict. So action of the radio, circular, and ciliary muscles. Viewing close object causes accommodation. The lens to change shape. External muscles control eye movement to follow objects. Viewing close objects causes convergence. Eyes moving medially. So talk about some pathology of the eye. So emmetropia is when the eye focuses images correctly on the retina. Okay, so that's normal. Emmetropia is normal. The eye is focusing images correctly on the retina. Myopia, okay, so this is nearsighted. So, and the way um, that it works, the name tells you what you can see. So myopia is nearsighted, meaning that you can see objects closely. You can't see distant objects. So distant objects appear blurry. Light from those objects fails to reach the retina, and a focus in front of it results from an eyeball that is too long. So myopia, you are nearsighted, meaning that you can see objects that are close. Objects that are far away, you cannot see. Okay. Hyperopia, you are farsighted, meaning that you can see objects far away, fine, but near objects are blurry. Okay, so near objects are blurry while distant objects are clear. Distant objects are focused behind the retina, result from an eyeball that is too short. So, so this is showing you the normal emmetropic eye right here. You see that the focal point falls nicely on the retina. And you see right here at B, the myopic eye. You see how the eye is too long. So you see how the focal point for the myopic eye does not fall on the retina, is in front. Okay, the eyeball is too long. It doesn't focus on the retina the way it's supposed to. And then you see the hyperopic eye. Okay, so the eyeball in this case is too short, and you see how the focal point is behind the retina, so it's not falling on the retina either. It's behind the retina. But you can correct nearsighted and farsighted with lenses. Okay, so a concave lens can correct the myopic, can correct nearsightedness. Okay, you cause it to fall, the focal point to fall correctly on the retina, and then a convex lens can correct farsightedness, which hyperoptic eye, and you can get the focal point to fall correctly on the retina. All right, so night blindness, remember, it's your rods. So remember, your rods is what allow you to see at night, and your rods don't have any color. Okay, the color comes from the cones, and the cones function in bright light. The cones function in bright light. It's the rods that function in the dark. So if you have night blindness, then um, you have inhibited rod function that hinders the ability to see at night. And color blindness is actually a genetic condition that results in the inability to see um, certain colors. And usually color blindness is in males because it's on the X chromosome. So because males only have one X chromosome, if they inherit a color blind allele, then it's going to be expressed. And then cataracts, when the lens become hard and opaque, our vision becomes hazy and distorted. All right, so now we are going to start talking about um, the ear and hearing and equilibrium. Okay, so the ear is divided into three major areas: the external ear, the middle ear, and the internal ear. The external and middle ear structures serve the needs of the hearing only. So hearing only is going to be with the external and middle ear, whereas the internal ear structure is going to deal with both hearing and equilibrium. So equilibrium is going to be dealing with the um, inner ear, and the inner ear also does still deal with hearing, but equilibrium is only going to be dealing with the inner ear. So the ear has receptors or mechanoreceptors, and different organs house receptors for each sense. So the external ear is called the outer ear. The middle ear, um, tympanic, has a tympanic cavity, and then the inner ear has the bony labyrinth. And this is a figure that shows you the external ear, the middle ear, and the inner ear. So again, the external ear, essentially the purpose of the external ear is to channel the sound waves um, to the middle ear, and then the middle ear is going to transmit it to the inner ear. But you have the auricle, all right, which is made out of elastic cartilage. So the auricle is going to channel the sound to the external acoustic meatus, and essentially the external acoustic meatus is going to transmit the sound waves from the auricle to the tympanic membrane. Okay, tympanic membrane right here, and the tympanic membrane is going to separate the external ear from the middle ear, and it's going to vibrate, okay, at the same frequency as the sound waves hitting it, and then it's going to transmit those vibrations to the inner ear, and you have your malleus, your auditory ossicles, which are your malleus, incus, and stapes. Okay, so and then you see this the malleus essentially looks like a hammer. Okay, incus looks like an anvil, and the stapes look like a stirrup. Okay, so the waves are going to transmit from the malleus, incus to the stapes, um, and then after it goes transmit um, to the stapes, then it's going to go to from the oval window, which you see right here, um, oval window connected right there to the stapes. So then the vibrations of the stapes at the oval window is just going to essentially create pressure waves that's going to be eventually transferred to the endolymph and the cochlear duct. And then in the cochlear duct, you're going to have hair cells, okay, which are going to bend in response to the um, vibrations, and that's going to transmit the sound waves to cochlear nerve. And then from the cochlear nerve, it's going to go to the auditory centers in the brain, in the temporal lobe of the cortex. So this is the cochlea right here, which has the hair cells are going to be in there. And then this, as far as the equilibrium, is going to deal with the vestibule, which is right here in the semicircular canals. Okay, so the semicircular canals and the vestibule are going to be the part of the inner ear that deal with the equilibrium. Okay. So the external ear, like I said, it's made out of elastic cartilage, the auricle, and is involved in hearing only. The middle ear, um, tympanic cavity, is also involved in hearing only. And you have the auditory tube connecting the middle ear. You have an auditory tube collecting the middle ear with the throat. If we go back to the figure, um, so it allows for equalizing pressure during yawning or swallowing. If we go back to the figure, you can see that auditory tube right here, okay, connecting the middle ear to the throat, and that can be used to equalize pressure. Um, all right, so bones of the middle ear, you have your three bones that span the cavity, the ossicles, malleus, incus, stapes, okay, and essentially the function, they just transfer the vibrations from the eardrum to first the malleus, then the incus, then the stapes. Then the stapes is going to transfer it to the oval window. Okay, so this is showing you the tympanic membrane right there. Then you see the malleus, incus, and stapes. Okay. And again, all they're doing, remember the sound waves hit the tympanic membrane, then those vibrations are transferred from the tympanic membrane to the malleus, which transfers to the incus, and then transfers to the stapes. Then the stapes is going to transfer to the oval window. So the inner ear or bony labyrinth includes sense organs for both hearing and balance. So remember, has hearing and balance, the inner ear. Filled with perilymph, similar to cerebrospinal fluid, contains a maze of bony chambers within the temporal bone. So you have your cochlea, which is going to deal with the hearing, and then you have your vestibule and your semicircular canals, which are going to deal with equilibrium. All right, so this is just showing you the semicircular canals, showing you the oval window, and it's showing you the cochlea right here, and then the vestibule is right here. All right. So the mechanism of hearing, so vibrations from sound waves move the tectoral membrane. All right. So hair cells are bent by the membrane, and action potential starts in the cochlear nerve, and impulses travel to the temporal lobe. So to kind of summarize um, this slide, so essentially the vibrations from, remember, the tympanic membrane go to the malleus, incus, and stapes, go from the stapes to the oval window, all right, which is going to create pressure waves in the perilymph, which is going to get transferred to the endolymph in the cochlear duct, and then in that cochlear duct, you're going to have your hair um, hair cells, okay, and then when they move, it's going to um, create that action potential, which is going to travel through the cochlear nerve to the auditory center in the temporal lobe of the cortex. All right, so equilibrium receptors again in the inner ear are called the vestibular apparatus. So it has two functional parts: static equilibrium and dynamic equilibrium. All right, so this figure is showing you static equilibrium. So essentially, static equilibrium is going to take place in the vestibule. All right, so in the vestibule, you have a different set of hair cells, okay, which are going to be, in this case, receptors that monitor head position and acceleration in a straight line. Okay, so these hair cells are going to monitor head position and acceleration in a straight line, and they are going to respond to gravitational pull. Okay, and that's going to provide information on which way is up or down, as well as changes in linear speed. So you see right here, you have, these are the hair cells, okay, and then you have this otolithic membrane, okay. So this otolithic membrane is a gelatinous material containing small grains of calcium carbonate, which are the otoliths. So you see these otoliths, which are essentially calcium carbonate. So what happens is when the head moves, all right, so you see right here in this figure, the head is moving. The otoliths, okay, those otoliths are going to move in response to variation in gravitational pull. And they're going to, when they do that, when they move, they're going to deflect different hair cells that trigger either hyperpolarization or depolarization of the hair cells and modify the rate of the impulses along the vestibular nerve, okay? And this figure is showing you dynamic equilibrium, and dynamic equilibrium is going to take place in the semicircular canals. All right, and they monitor rotational acceleration. So essentially, at the base of each of the semicircular canals is an enlarged region, the ampulla. So you see right here, this would be the base of a semicircular canal. You have this ampulla, okay? You have this enlarged region, and then within the ampulla is a receptor region called the crista ampullaris, okay? This is the crista ampullaris, all right, which consists of a tuft of hair cells covered with a gelatinous cap, all right? So you see your hair cells covered with a gelatinous cap, okay? The culpa of the crista, that gelatinous cap is the culpa of the crista ampullaris, okay? So during rotational acceleration, when you begin to twirl around, the endolength in the canals are going to lag behind the head, the actual head movement, because of inertia, okay? All right, so essentially what happens is the head movement is going to depolarize the hair cells as a result, and the impulse gets transmitted through the vestibular division of the cranial nerve 8. So then what happens when the rotation movement suddenly stops? The endolength is going to keep going in the same direction the head was moving, again due to inertia. And this is going to push the ampullary culpa, essentially those hair cells, in the same direction as the previous head movement, and it's going to cause it to hyperpolarize the hair cells All right, and when the hair cells are hyperpolarized, they aren't going to transmit any signals. Okay, so that essentially tells the brain that you have stopped moving. All right, so with dynamic equilibrium, again, it deals with angular head movements. The movement of the culpa stimulates the hair cells, and the impulse is sent via the vestibular nerve to the cerebellum. All right, so this is a good table that summarizes the internal ear, the function, and then where the receptors are located. So the semicircular canals, all right, it deals with equilibrium, all right, rotational acceleration, and you have the hair cells in the crista ampullaris, and then the vestibule. Um, so you find it deals with also equilibrium, and it deals with static equilibrium and head position relative to gravity in the linear acceleration, and it's in the receptors in the macula, and then with hearing, with the cochlea, it deals with hearing, okay, and the receptors in the spiral organ. All right, so now dealing with taste and smell. So they both use chemoreceptors. So they're stimulated by chemicals in solution. Taste has 4 types of receptors. Smell can differentiate a large range of chemicals. Both senses complement each other and respond to many of the same stimuli. If you notice, a lot of times when you get sick and your nose gets stuffed up, your sense of taste also goes due to your stuffed up nose. So smell and taste um, complement each other. So you have olfactory receptors in the roof of the nasal cavity. So neurons with long cilia, and then chemicals must be dissolved in mucus for detection. Okay, and impulses are transmitted via the olfactory nerve, and interpretation of smell is made in the cortex. And this is showing you the olfactory bulb, and then it's showing you the how within the smell has to be dissolved in the mucus layer, okay, and then you have these supporting cells with olfactory receptors that can sense those that diffuse chemicals and then transmit a signal, okay. Um, so you see at the end of these olfactory receptors, you have those olfactory, essentially, um, cilia, all right, and it's those cilia that's going to detect those that diffuse chemicals in the mucus layer. All right, so taste buds house the receptor organs. Location of taste buds, most are on the tongue. You have some on the soft palate and the cheeks. All right, so the tongue is covered with projections called papillae and filiform papillae, sharp with no taste buds, fungiform papillae, rounded with taste buds, and circumvallate papillae, large papillae with taste buds. All right, so taste buds are found on the sides of the papillae. All right, so these are showing you right here the fungiform papillae, and then you see some other structures of the mouth. The epiglottis. Epiglottis essentially covers the windpipe when you eat and swallow. That's what the epiglottis does. Then you have your palatine tonsils and lingual tonsils. So these are showing you the cranial nerves involved. So you have facial nerve, which is cranial nerve 7. You have the glossopharyngeal nerve, which is cranial nerve 9. Okay. And then you have the vagus nerve, all right, which is, and it also shows you the structures is innervating. So the vagus is going to the epiglottis. Facial nerves are going to the more tongue area, facial nerve, innervating the tongue area, and then the glossopharyngeal nerve is innervating more of the pharyngeal area. So you have sweet receptors, sour receptors, and bitter receptors, and salty receptors. So different receptors of the tongue detect different, pretty much tastes, whether sweet, sour, bitter, or salty. So conclusion: special senses, smell, roof of the nasal cavity, taste with the taste buds, and sight. 70% of all sensory receptors are in the eyes, and then you have with the ears, you have the hearing in equilibrium. All right, so that is the end of the notes for chapter 15.